All NEET aspirants, you are listening to podcast Botany for NEET by S. Ramakrishna. In this podcast, recombinant DNA technology, the tools of recombinant DNA technology. So, for successful recombinant DNA technology, the key tools which are so important: restriction endonucleases, what we call molecular scissors, molecular knives, molecular scalpels, and DNA ligase, what we call molecular glue or molecular stitches, and DNA polymerases, the enzymes which polymerize deoxynucleotide to make DNA, and vectors to carry the foreign DNA to host cell, and finally the host cell, which is considered as expressing system to express the foreign gene to produce a particular protein. Next, the first one, restriction endonucleases. As the word itself conveying its meaning, it restricts the multiplication of viruses inside the bacterial cells, that is E. coli. So E. coli have a defense mechanism to prevent the viral multiplication, to restrict its multiplication inside the bacterial cells. So it is having a modification enzyme system, because the E. coli, it is having restriction endonucleases, it is going to cleave the DNA, but its own DNA got protected because this enzyme unable to recognize its own DNA, the bacterial DNA, because the bacterial DNA, it got methylated. The enzyme which adds methyl groups to the host DNA, what we call methylase. So as the DNA got methylated, not recognized by the restriction enzyme, this enzyme recognize the viral DNA that is unmethylated. So because the enzyme having specificity, as it recognizes unmethylated DNA, the viral DNA, it cleaves the viral DNA into fragments. So viral multiplication prevented inside the bacterial cells. So methylase and other it is restriction enzyme. Next, the first restriction enzyme discovered was HIND2 from Haemophilus influenza, a bacteria, and it is having a special recognition sequence of 6 base pair GCTAGC. The enzyme breaks between T and A, that is thymine and adenine, pyrimidine and purine, from 5' to 3' and other strand the 5' to 3'. It results in blunt ends, non-sticky end, double strand end, because exactly cleave the DNA across the DNA. So it results in double stranded DNA, non-sticky ends. Next, more than 900 restriction enzymes are discovered from over 230 strains of the bacteria. So restriction enzymes are present only in case of the prokaryotes, the bacteria. Next, E-core1, that is from Escherichia coli RY13 bacterium. In case of E-core1, the E-core1, the first letter, that is the capital letter, indicates the genus. The second two letters, CO, indicate the species, and R indicates the name of the strain, and Roman 1, it is order of isolation from that particular bacterium E. coli. Earlier we said HIND2. Suppose HIND1. HIND1 was not discovered first, but it isolated first from Haemophilus. HIND2, it was discovered first, but isolated second from that particular bacterium Haemophilus. Here also, E-core1, it extracted the first from that bacterium, that is Escherichia coli. Next, the restriction endonucleases belongs to large class of enzyme. What we call it is nucleases. The nucleases belongs to third class according to IUB, that is hydrolases. And nucleases are the two types, exonuclease, endonuclease. Exonuclease, the word itself conveying its meaning. It acts outside the DNA having free ends. That exonuclease do not act on circular DNA because circular DNA do not have free ends. Exonuclease cleaves the DNA into fragment from outside to inside. In case of endonuclease, the endonuclease, here also the word conveying its meaning, within the DNA. As restriction endonuclease having special cuts, the precise cuts in case of the DNA. Because restriction endonuclease are endonucleases, but all endonucleases are not restriction endonucleases. The endonuclease cuttings which are random, but restriction endonuclease cutting are specific and precise. Next. The restriction endonucleases first inspect the length of DNA sequence. Usually it is the 6 base pair length, that is the 12 nitrogen bases. Find the recognition sequence. The restriction enzyme having a recognition sequence generally in the palindrome form. A palindrome is nitrogen bases read same in both the forward and backward direction. After recognition, it binds to it and cleaves the DNA in a particular fashion. In case of palindrome, if you take the word palindrome, Malayalam, it reads same both forward as well as backward direction, but not in case of nucleic acid, the double-stranded DNA. It is mirror image palindrome. In case of 5-3 and 3-5, the orientation of reading must be kept same. In 5-3, GAA TTC. In case of second strand, opposite strand, GAA TTC from 5 direction. Suppose the 3, CCT AAG. Here also CCT AAG. So the sequence should be 5-3 in 5-3 or 3-5, otherwise it is 3-5. Next. The restriction enzyme, in case of E-core1, it cleaves the DNA, symmetric cleavage of phosphodiester bond on both strands at different location between same nitrogen bases, just mid away from the palindrome. GAA TTC, it breaks between G and A from 5 to 3 direction. That is purine, purine. In case of second strand, 5 to 3, G and A. So, symmetric cleavage, phosphodiester bond, just mid away from the palindrome, results in single-stranded sticky end at the end of double strand. The single-stranded sticky ends, it facilitates the activity of DNA ligase. If you use the same restriction enzyme in source DNA and vector DNA, the sticky ends are formed, and sticky ends are ligated from one end to other end. It results in a recombinant DNA molecule. Suppose if you use the A restriction enzyme in source, another B restriction enzyme in vector. In source and vector, sticky ends are formed, but the sticky ends are not matching. That is not complementary. So, unless you use the same restriction enzyme in source and vector, recombinant molecules are not expected. You have to use the same restriction enzyme, source and vector, then only they are ready complemented and form the hydrogen bond of the sticky ends. Next, as the DNA fragments generated by restriction endonucleases separated by gel electrophoresis. The gel, that is agar gel obtained from red algae member, Gracilaria and Giridium. It is a natural polysaccharidic gel which providing sieving effect. So a molecular sieve is, that is agar gel. Electrodes are used in the medium or matrix to push the DNA towards anode. As it is negatively charged, we push towards what it is anode positively charged. Phoresis to resolve, to separate based on charges and sizes. More appropriately, charges, because net charge is going to be negative only. Next, in case of diagrammatic representation of agarogel electrophoresis, first, second, third, fourth line. First line it is undigested line. Second, fourth line it is the digested line. Smaller the fragment, farther away from gel well towards anode. Larger the fragment, farther away from anode towards it is the gel well. So, speed of the fragment related to size of the fragment. So, size of the fragment is inversely related to the speed. So, speed decreases means it is size increases. As size decreases, speed increases towards anode. Next, these fragments present in case of the gel are not visible in case of the visible light, because DNA is colorless. It absorbs ultraviolet radiation only, that is the 240, 250 nanometers of wavelength of light. That is not in case of visible light range as 400 to 700 nanometer. So it is not visible. So it should be stained with ethidium bromide, a fluorescent dye. It is intercalated between nitrogen bases. Even then it is not visible. It is exposed to ultraviolet radiation. So the fluorescent dye gives orange color. That is in case of visible range. Then DNA it is detected. So DNA detected because of appearance of bright orange colored bands. The bright orange colored bands appear only after treatment with, that is using dye ethidium bromide and exposed to ultraviolet radiation. And the gel having bright orange colored band cut into pieces. From each piece, the fragment of DNA extracted. So we are separating the fragment of DNA from other molecule. What do you call it is elution. Now eluted DNA ready to link with the vector to carry this to host cell for multiplication of this desired gene. Suppose in case of restriction NJ, it is E core 1, it is having the sequence GAA-TTC at 4 points in linear DNA, double-stranded DNA. So it is going to generate 5 fragments, that is n plus 1, the formula. 4 points, recognition sequence, generating 5 fragments. In 5 fragments, the last 2 fragments, which are single strand at the end of double strand, last 2 fragments. In middle 3 fragment, single strand on either side of the double strand. It is one single strand, it is one single strand, middle it is a double strand. 3 fragments are generated like this. So n plus 1 applicable to linear double stranded DNA. Suppose if we take the circular DNA, the same E core 1 at 4 points, GAA TTC. In this, the principality is n is equal to n. There are 4 cleavage points. That means 4 fragments are generated in case of circular DNA. All these 4 fragments are like this only. That is single stranded on either side of the double strand. In first case, in linear, the 2 fragments are single strand at the end of double strand. The rest of the fragments are single stranded on either side of the double strand. That is the difference. Next, vectors. Vector is a carrier of the DNA to the host cell for its multiplication. What do you call it is the cloning vector. To ferry foreign DNA to host cell. What do you call cloning vector? Phases, that is bacteriophages and plasmids, are used as vector. The phases and plasmids which multiply inside the bacterial cell independent of the genome, that is the coding strand. And phases having high number per cell equal to copy number of the genome of the phase. Suppose the phase, after multiplication in each bacteria produce 100 viral particles. That means if you link the foreign DNA, you are going to get 100 copies. So it is the 100, it is the copy number of that phase. In case of the plasmid, it is having 1 to 2 copy, 10 to 15, 100, even more than 100. So if you link the foreign DNA to phase or plasmid, it multiplies and gives the number of copies equal to copy number of phase and plasmid. Suppose this plasmid inside the bacterial cell, it produces suppose 50 plasmid. So if you link the foreign DNA to plasmid, you are going to get the 50 copies in each bacterial cell. That 50 is the copy number of the plasmid. So if you link the foreign DNA to phase DNA or plasmid DNA, multiply, and this multiplied number is equal to copy number of phase, copy number of plasmid. So vectors constructed in such a way that vectors are engineered in such a way that easy linking of the DNA, that is one thing, and selection of recombinants from non-recombinant. We are going to select only recombinant. We have to eliminate non-recombinant. That is one. Next, the characteristic features of vector. First, ORI. The ORI is a sequence of nitrogen bases in case of the vector DNA, where initiation of DNA replication occur. Not only this, ORI is also responsible for controlling the copy number of linked DNA. Suppose it is a vector, it is having ORI. It is the foreign DNA. So it is going to decide the foreign DNA, how many number it should be. So ORI of the vector is going to determine the copy number of linked DNA. So we should prefer the vector having ORI with high copy number, it is preferred. Next, selectable markers. Suppose it is a vector, it is the foreign DNA. If you want to link this into this vector can accept this, and other vector it cannot accept this. If both vectors are allowed for transformation to the bacterial cell, some bacterial cell can accept this vector, some bacterial cells cannot accept this vector. So transformed cells are formed, non-transformed cells are formed. And we have to select the only transformed cell, eliminate non-transformed cell. So for selectivity of the transformed cell, we must have certain markers. What do you call it? Is selectable markers. The selectable markers are antibiotic resistant genes, not antibiotic producing gene. And normal E. coli do not have any of the resistance to these antibiotics like ampicillin, tetracycline, kanamycin, chloramphenicol. So how? It should be restructured, it should be modified in such a way that it should be with antibiotic resistant gene. In such a way, the plasmids which are modified in laboratories, which is used as a vector, in that one, plasmid Bolivar Rodriguez. The other one may be plasmid University of California. So PUC, PBR, plasmid, BR, Bolivar, or for Rodriguez 322. That is order of preparation in that particular laboratory. PBR 322. If you look at the diagram in case of the text, PBR 322. PBR 322 is a polynucleotide. So it is having on top ECOR1, CLAV1, HIND3. It is HIND3. In right side, it is having tetracycline resistant gene. In left side, ampicillin resistant gene. In tetracycline resistant gene, BOMH1, SOL1. In case of ampicillin resistant gene, PST1, PVU1. In PBR322, there is a ROP, ROP. that is gene encoding for protein required for replication of this plasmid, that is the rope, and to this side origin of replication. So E core 1, clav 1, Hind 3, bomb H1, sol 1, PST1, PVU1, and at row PVU2. So 3 plus 2 plus 2 plus 1. So 9 point, the 3 points at the top, and two, that is two restriction enzymes at side. So cloning sites which are restriction site. So all cloning sites are restriction site, but all restriction sites are not a cloning site. You will see this. So E core 1, clav 1, Hind 3, bomb H1, sol 1, PST1, PVU1, PV2, which are all are polypeptide, which act on polynucleotide, that is PBR 322. Next, cloning site. Earlier we said all cloning sites are restriction site, but all restriction sites are not a cloning site, because at a rope PVU1 we cannot ligate the foreign DNA at a rope. Even though we ligate, the process is going to be futile, because the gene encoding for protein is required for replication of this plasmid. Without replication, it is not successful. So even though if you ligate the DNA at the rope, it is not going to be successful. So all cloning sites are restriction site, but all restriction sites are not a cloning site. So in cloning site, it should have few restriction site. For commonly used restriction enzyme, preferably one site for one enzyme. Suppose E core 1, it should be one site only. If it is each restriction enzyme having more than one site, it makes the process highly complicated. It makes the DNA into fragment. Some useful points are going to be eliminated. Suppose A, B, C restriction enzyme. A must be with one site, B must be with one site, C must be with one site. Suppose A having 4 site, it is not ideal. B having 3 site, it is also not ideal. So A restriction enzyme one site, B other restriction enzyme one site, C for one site, it is ideal. So preferably one site. In this, the point is all cloning sites are restriction site, but all restriction sites are not a cloning site. Next, its molecular weight. The vector molecular weight is generally should be low, because providing deoxynucleotide enzymatic machinery on the part of the host is going to be a metabolic burden. And in order to retain the vector, the foreign DNA inside the cell, it must be with antibiotic resistant, because otherwise the host tend to lose what it is, the plasmid. Next, so we can ligate the foreign DNA at any one of the antibiotic resistant gene in case of PBR 322. Suppose if you ligate the foreign DNA by using bomb H1 and a DNA ligase. So as you are using bomb H1 and for ligation DNA ligase, in case of PBR 322, it will lose tetracycline resistant gene. It is having ampicillin resistant gene. So even though it is without tetracycline resistant gene, it can be identified by ampicillin resistant gene. First, it is incubated in a medium having the antibiotic ampicillin, then tetracycline. The recombinant can able to grow in case of ampicillin, but unable to grow in case of tetracycline, because it lost tetracycline resistant gene, unable to grow in tetracycline. Non-recombinant can grow in both ampicillin as well as tetracycline, because for ligation of the foreign DNA vector, how many types of vector are formed? 2 types of vector. One, it is recombinant vector. Other is non-recombinant vector. In this non-recombinant vector, it is having ampicillin resistant gene, tetracycline resistant gene. In this, it is having only ampicillin resistant gene but not tetracycline. The two vectors allowed for transformation with bacteria. 1, 2, 3. Three kinds of cells are formed. A, B, C, suppose. A transformed with recombinant. B transformed with non-recombinant. C non-transformed cell. So three kinds of cells are generated. So in first one A, the recombinant is able to grow. In second one, recombinant unable to grow. Non-recombinant can grow both in case of ampicillin as well as what it is tetracycline. So non-transformed cells will not survive. So one antibiotic resistant gene, that is ampicillin resistant gene, help in the selection of transformant. Other antibiotic resistant gene, that is tetracycline, which got inactivated due to insertion of the foreign gene, helps in the selection of recombinant. So selection of recombinant by insertion of the foreign gene at tetracycline resistant gene and such insertion inactivation and selection of recombinant is a cumbersome process that is unmanageable. Why it is a cumbersome, that is unmanageable? Simultaneous plating on both antibiotics required, ampicillin as well as tetracycline. So it is cumbersome. He has to prepare the master plate. He has to see this, how many recombinant colony, non-recombinant colony, and again he has to make to grow in case of ampicillin, tetracycline. Such method, it is a cumbersome, unmanageable. You are not saying not possible, but it is unmanageable. So alternate selectable markers are discovered. That is ability to produce color in presence of a chromogenic substance. That is X-gall. The X-gall is a substance or maybe dye. It is hydrolyzed by beta-galactosidase. So beta-galactosidase hydrolyze X-gall into some color, the blue color. So within the recombinant DNA, if we want to ligate the foreign DNA, a lag-Z gene site. Suppose in case of the plasmid, if we insert the foreign DNA, a lag-Z gene site, it will lose lag-Z gene. So there is no product for transcript for lag-Z gene because already it lost. No translation, no protein. In that medium, X-gall is there. So X-gall is not hydrolyzed by beta-galactosidase. Why there is no beta-galactosidase? Due to insertion inactivation of the gene encoding for beta-galactosidase due to insertion of the foreign gene. As we inserted the foreign gene, it loses lag-Z gene. There is no transcript of lag-Z gene. There is no protein product, translation product, that is beta-galactosidase. So the X-gall remain as it is, that is colorless. So colorless colonies which are recombinant colonies. So plasmid DNA disrupted with the foreign gene. So it is plasmid DNA not intact in first one. In second one, within the plasmid DNA, we want to insert the foreign DNA, a lag-Z gene. It do not accept this. So lag-Z gene is intact. Lag-Z gene produce what is transcript mRNA. Translation produce beta-galactosidase. So beta-galactosidase in this case of medium, which will hydrolyze X-gall into blue color. So blue colored colonies are non-recombinant, white colored colonies are recombinant. So within the petri dish, with the color we can able to recognize which is recombinant, which is non-recombinant. So in case of non-recombinant, the plasmid, it is intact plasmid, that is lag-Z gene present. In case of recombinant colony, in case of the plasmid, lag-Z gene absent. So with the color we can identify which is recombinant, which is non-recombinant. What we call insertional inactivation of the gene encoding for beta-galactosidase. In earlier, Insertion inactivation of the gene encoding for antibiotic resistance. In this gene encoding for beta galactosidase. So LAGZ gene beta galactic code for beta galactosidase which hydrolyze, that is X-gall, into the colored substance. So recombinant colonies are colorless. Non-recombinant colonies are blue colored. Next, vectors. Since right from the beginning, we are so familiar with bacteria, virus, transfer the genes to plant, animals, and direct the plant animal cell to synthesize the chemicals as required by the bacteria and the virus. So it will mediate the host cell to synthesize the chemical for its survival, the bacteria and virus. If you able to understand the art of delivery of the genes by bacteria and virus to the plant and animal cell, certainly it generates the knowledge to modify the tools, which tools are responsible. If you modify the tools, we can use it as a vector to deliver the gene of interest into plant and animal cell. First one, Agrobacterium tumefaciens, that is a natural genetic engineer. This Agrobacterium tumefaciens having Ti plasmid, tumor-inducing plasmid. It is pathogen for several dicot. Normal cells are converted into tumor cell. In other, retrovirus, which is converting normal cells into cancer cell. Both are pathogenic, so it should be made non-pathogenic. So Agrobacterium Ti plasmid should be modified. Whatever the segment tDNA, tumor-inducing sequence, that is responsible for conversion of normal cells into tumor cells, that should be deleted. Suppose it is a vector Ti plasmid, it is a tool. It is the tDNA. tDNA got deleted. We inserted the foreign DNA. Now this vector is no more a pathogenic, a disarmed vector. So the disarmed vector with the foreign DNA used to deliver the gene of interest into variety of the plant because it is disarmed vector. Like in case of animal cell, retrovirus disarmed to deliver gene of interest into animal cells. Next, competent host. As the DNA is hydrophilic, it cannot pass through the cell because the membrane it is hydrophobic. DNA hydrophilic, the membrane it is hydrophobic, it cannot pass through it. So we had to increase the efficiency of the cell, bacterial cell, to accept the foreign DNA, that is recombinant DNA. How can you increase the efficiency of the bacterial cell? By treating with specific divalent calcium ion, Ca+2, that is calcium chloride solution. It makes the transient pores on the cell wall. So in bacteria, transient pores are found first on the cell wall. So foreign DNA has to force to enter into the bacterial cell by incubating this first on ice, then followed by 42 degrees centigrade heat shock, then ice. So ice, heat shock, ice method is applicable in case of the bacteria. So now recombinant DNA forcefully enter into the bacterial cell. So first treated with divalent calcium ion, specific divalent calcium ion, then incubated on ice, heat shock and ice. Now bacterial cells can take the foreign DNA, that is recombinant DNA. It is applicable in case of bacteria. In case of animal, micro injection of the recombinant DNA directly into nucleus in case of animal cells. Next, in plant cell, a high velocity micro particle, the tungsten and the gold, the high velocity particle, it is coated with foreign DNA, and by using gene gun or shotgun, the plant cells are bombarded with the high velocity particle. The plant cells can take up foreign DNA. Since plants which is applicable, gene gun shotgun method, or biolistic method. Which particles are used? High velocity particle like tungsten and gold coated with foreign DNA. Next, the other last it is disarmed vector method. Agrobacterium in plants, retrovirus in case of animals. I am signing off from this episode. You will see in case of the next episode, that is process of recombinant DNA technology. So listen with concentration, connect to the dots of the concept, feel easiness to recall at the time of examination. If you feel this approach is right, don't forget to subscribe and share to the person those seriously preparing for NEET.